Dear friends, today we are going to discuss pedigree and probability. First, the main objectives of the topic are: the first objective is how pedigree analysis can provide insight into the genotype of relatives based upon the phenotypes observed. Number two, to construct a pedigree based on family history. Then, to analyze the information in pedigree by applying the laws of inheritance. Then, to determine the mode of inheritance of traits based upon the information in pedigree. The last objective of the topic is the probability a child of particular parents would inherit the trait in question. Before going into detail, let us have a brief summary of the topic. First, the study of human genetics is complicated because controlled matchings in humans are not possible for ethical reasons. The inheritance patterns of human traits usually are identified by examining the way the traits occur in family trees of individuals who clearly exhibit the trait. An important technique used by geneticists to study human inheritance is the analysis of pedigree, and for determining the outcome of genetic cross, the rules of probability are used. Now, what probability actually is? Probability expresses the likelihood of the occurrence of particular event, here the occurrence of a genetic disorder. It is the number of times that a particular event occurs divided by total number of all possible outcomes. Suppose we conduct a simple statistical experiment. We flip a coin in one time. The coin flip can have one or of two equally likely outcomes, heads or tails. Together, these outcomes represent the sample of our experiment. Individually, each outcome represents a sample point in the sample space. What is the probability of each sample point? The sum of probabilities of all the sample points must be equal to 1. And the probability of getting a head is equal to the probability of getting a tail. Therefore, the probability of each sample point, head or tail, must be equal to half. Before going in detail, let's have a cursory look of symbols that are usually used in pedigree analysis. The symbols commonly used in pedigree are: males in pedigree are represented by squares, females by circles. A horizontal line drawn between two symbols represents a man, and a woman indicates a mating. Children that are connected to their parents by vertical lines extending downward from their parents. Persons who exhibit the trait of interest are represented by filled circles and squares. Unaffected members are represented by open circles and squares. The person from whom the pedigree is initiated is called proband and is usually designated by an arrow. Now, going into the detail of the topic, let us start from autosomal recessive traits. Autosomal recessive traits appears in both the sexes with equal frequency. Traits tend to skip generations. The affected offsprings are usually born to unaffected parents. When both parents are heterozygous, approximately 1/4 of the progeny will be affected. Appears from frequency among the children of consanguineous marriage. A number of human metabolic diseases are inherited as autosomal recessive traits. Important among these is albinism. Albinism means deficient in pigmentation. Individuals with albinism do not produce the pigment melanin, which protects the skin from harmful UV radiations or ultraviolet radiations. Consequently, their skin and eyes are very sensitive to sunlight. Frequencies of harmful recessive mutant alleles usually are higher than frequencies of harmful dominant mutant alleles, because heterozygotes for the recessive mutant allele are not at a significant selective disadvantage. Nonetheless, individuals heterozygous for harmful recessive mutant alleles usually are rare. In the United States, approximately 1 among 17,000 of white population and 1 among 28,000 of American population have albinism. Now about autosomal dominant traits. Autosomal dominant traits appear in both sexes with equal frequency. Both sexes transmit the trait to their offsprings. Autosomal dominant traits do not skip generations because they are dominant. Affected offsprings must have an affected parent unless they possess a new mutation. When one parent is affected, heterozygous, and the other parent is unaffected, approximately half of the offsprings will be affected. Unaffected parents do not transmit the trait. If an autosomal dominant allele is rare, most people displaying the trait are heterozygous. When one parent is affected and the heterozygous, then the other parent is unaffected. Approximately half of the offsprings will be affected. If both parents have the trait and are homozygous, approximately 3/4 of the children will be affected. Unaffected people do not transmit the trait to their descendants, provided that the trait is fully penetrant. A trait that is usually considered to be autosomal dominant is an example of hypercholesterolemia, an inherited disease in which blood cholesterol is greatly elevated owing to a defect in cholesterol transport. Cholesterol is transported throughout the body in small soluble particles called lipoproteins. A principal lipoprotein in the transport of cholesterol is low density lipoprotein designated as LDL. When an LDL molecule reaches a cell, it attaches to an LDL receptor which then moves the LDL through the cell membrane into the cytoplasm, while it is broken down and its cholesterol is released for the use by the cell. Familial hypercholesterolemia is due to a defect in the gene that normally encodes LDL receptor. The disease is usually considered an autosomal dominant disorder because heterozygotes are deficient in LDL receptors and have elevated blood levels of cholesterol, leading to increased risk of coronary artery disease. Persons heterozygous for familial hypercholesterolemia have blood LDL levels that are twice normal and usually have heart attacks by the age of 35. Very rarely, a person inherits two defective alleles for the LDL receptor. Such persons do not make any functional LDL receptors. Their blood cholesterol levels are more than 6 times normal and they may suffer a heart attack as early as age 2 and almost inevitably by age 20. Because homozygotes are more severely affected than heterozygotes, familial hypercholesterolemia is said to be incompletely dominant. However, homozygotes are rarely seen and the common heterozygous form of the disease appears as a simple dominant trait in most pedigrees. Dear students, now let's discuss the X-linked recessive traits. In case of X-linked recessive traits, males are more affected than females. Number 2, affected sons are usually born to unaffected mothers, thus the trait skips generations. Approximately half of carrier mothers' sons are affected. It is never passed from father to son. All daughters of affected fathers are carriers. An example of X-linked recessive trait in humans is hemophilia, also called classic hemophilia. Hemophilia results from absence of protein necessary for blood to clot. Hemophilia is carried on X chromosome. It is called X-linked genetic disorder. A woman who is a carrier for hemophilia has the genetic mutation on one of her X chromosomes. She will have another non-mutated X chromosome that will usually compensate for the defect in the other. It is not uncommon for women who carry the hemophilia gene to have low levels of clotting factor and have bleeding problems. A man who has hemophilia has the genetic mutation on its only X chromosome. He does not have another X chromosome to compensate for the defect, so he will have hemophilia. 2/3 of the cases of hemophilia are inherited. The other 1/3 occur with no family history and are called spontaneous genetic mutations. The spontaneous genetic mutations may either occur in female fetus or a male fetus. The female fetus will become a carrier of hemophilia. The male fetus will become a person affected by hemophilia. There are 13 blood clotting proteins called coagulation factors found in the blood. They are designated by Roman numerals 1 through 13. When a blood vessel is damaged, these clotting factors are switched on in a certain order called blood clotting cascade and work to form a clot. Specifically, these 13 factors normally combine to a clot. If one factor is missing or present at low levels, this causes hemophilia and the other blood clotting problems, and a proper clot will not form. The two most common factor deficiencies are factor 8 and factor 9 deficiency. The most common affecting 80% of the hemophilia population, those with hemophilia A. Hemophilia A is factor 8. When these blood clotting proteins aren't present, is not easily stopped. Now let us discuss X-linked dominant traits. X-linked dominant traits, both males and females are affected. However, females are more affected than males. X-linked dominant traits do not skip generations because they are dominant. Affected sons must have an affected mother. Affected daughters must have either an affected mother or an affected father. Affected fathers will pass the trait on to all their daughters. Affected mothers, if heterozygous, will pass the trait on to half of their sons and half of their daughters. An example of X-linked dominant trait in humans is hypophosphatemia. X-linked hypophosphatemia, designated as XLH, is a hereditary renal phosphate wasting disorder characterized by hypophosphatemia, also called rickets or osteomalacia, and diminished growth. It is the most common form of hereditary hypophosphatemia with a prevalence of approximately 1/20,000th. The disease affects both sexes equally. X-linked hypophosphatemia manifests during childhood with typical clinical features of rickets such as short stature, bone pain, the skeletal deformities, for example, bowed legs, dental abnormalities, abscesses, cavities, abnormal enamel, and are observed in children and adults. Cranial anomalies are also observed due to thickness of parietal and frontal bones. In adults, osteoarthritis of the lower limbs, and in some rare cases, hearing loss has been observed. Muscle weakness and hypotonia are absent. The disease is caused by various mutations in the gene PHEX gene and is transmitted as an X-linked dominant trait with complete penetrance, but variable expressivity. PHEX encodes an endopeptidase expressed predominantly in bone and teeth that regulates fibroblast growth factor 23, designated as FGF23 synthesis through unknown mechanisms. PHEX mutations lead to increased circulating levels of FGF23, a phosphate regulating hormone phosphatonin that leads to reduced renal phosphate absorption and consequently abnormal bone mineralization. Dear students, now we are going to discuss Y-linked traits. Y-linked traits exhibit a specific, easily recognized pattern of inheritance. Only males are affected and the trait is passed from father to son. If a man is affected, all his male offsprings also should be affected. Y-linked traits do not skip generations. Maleness is one of the few traits in humans that have been shown to be Y-linked. Retinitis pigmentosum. This disease is caused by mutation in the RPY retinitis pigmentosa Y-linked gene, which causes it to make faulty versions of protein needed for normal vision. Retinitis pigmentosa is a genetically inherited disease of the eye in which the cells of the retina produce defective proteins. The symptoms include a progressive loss of sight starting with decreased night vision, followed by loss of peripheral or side vision, and finally blindness. There is no treatment or cure for the disease. Dear friends, now how probability is used in case of pedigree analysis. Let's make the use of probability in solving the pedigree problems by taking an autosomal recessive trait into consideration. A few basic facts about autosomal recessive traits will allow us to calculate the probability that unaffected individuals are carriers. An affected individual must be homozygous recessive and must pass one disease allele to each one of its children. An affected child must inherit one disease allele from its mother and one from its father. A parent with single disease allele has a probability of half of passing the allele to any one of its children. Dear friends, now how to calculate probabilities from these autosomal recessive traits. First, the probability that an individual is a carrier is a number between 0 and 1. If we know an unaffected individual must be a carrier, then the probability is 1 that the individual is a carrier. If we know an unaffected individual cannot be a carrier, then the probability is 0. That means individual is carrier. If we know that one parent is carrier, that means probability is equal to 1 and the other parent is not the carrier, then probability is equal to 0. We know there are two equally likely possibilities. Number 1, if the child inherits the first parent's single disease allele, the child will be a carrier. Now, if the child inherits the first parent's other allele, the child will not be a carrier. Since there are total of two equally likely outcomes, and in one of those two cases, the child is a carrier. The probability that the child is a carrier is half. Calculating the probability an unaffected child is a carrier when one parent is not a carrier. If one parent is not a carrier, then a child can only inherit a disease allele from other parent. In these problems, we can assume that any individual marrying into the family is not a carrier. As we have just discussed, if the other parent is definitely a carrier, then the probability of passing that allele to child is half. And so the probability the child is a carrier is also half. But suppose that the probability that the other parent is a carrier is only half instead of 1. Because the probability the child will be carrier is half plus half, that is 1/4. Here is why the probability the parent is a carrier is half. And if the parent is a carrier, the probability of passing the allele to a child is just half. The probability that the parent is a carrier and that the parent passes the allele is half times half, that is equal to 1/4. In general, if one parent is not a carrier, the probability that a child will be carrier is half times the probability the other parent is a carrier. That is, we multiply the probability of passing a disease allele half times the probability that the parent does in fact carry the disease allele. Now, how sum rule can be used to predict the occurrence of mutually exclusive events. Now we have an understanding of probability, we can see how mathematical operations using probability values allow us to predict outcome of genetic crosses. Our first genetic problem involves the use of sum rule, which states that the probability that one of the two or more mutually exclusive events will occur in equal to sum of the individual probabilities of the events. As an example, let us consider a cross between two mice that are both heterozygous for genes affecting the ears and tail. One gene can be found as an allele designated as DE, which is a recessive allele that causes droopy ears. The normal allele is DE. An allele of the second gene causes a crinkly tail. This crinkly tail allele designated as CT is recessive to normal allele CT. If a cross is made between two heterozygote mice designated as DE, CT, CT, the predicted ratio of the offsprings will be 9:3:3:1 with droopy ears and a crinkly tail. These four phenotypes are mutually exclusive. For example, a mouse with droopy ears and a normal tail cannot have normal ears and a crinkly tail. The sum rule allows us to determine the probability that we will obtain any one of two or more different types of offspring. For example, in a cross between two heterozygotes designated as capital DE and small de and capital CT, small ct crossed with capital DE, small de, capital CT, small ct. We can ask a question, what is the probability that an offspring will have normal ears and normal tail or have droopy ears and a crinkly tail? In other words, if we close our eyes and pick an offspring out of a litter from this cross, what are the chances that we would be holding a mouse that has normal ears and normal tail or a mouse with droopy ears or a crinkly tail? In this case, the investigator wants to predict whether one of the two mutually exclusive events will occur. A strategy of solving such genetic problems using sum rule is described here. If we cross two homozygotes, the question can be asked like this: what is the probability that an offspring will have normal ears and normal tail or have droopy ears and a crinkly tail? The steps involved during this problem, we can calculate the individual probabilities of each phenotype. This can be accomplished using a Punnett square. The probability of normal ears and a normal tail is 9/16. The probability of droopy ears and a crinkly tail is 1/16. Now step number 2. Add together the individual probabilities, we will get 9/16 plus 1/16, that is 10/16. This means that 10/16 is the probability that an offspring will have either normal ears and a normal tail or droopy ears and a crinkly tail. We can convert this 10/16 to 0.625, which means that 62.5% of the offsprings are predicted to have normal ears and normal tail or droopy ears and a crinkly tail. With these words, we will conclude the topic. Thanks for watching.